Hi, I'm Zhenghong from Case Western Reserve University, Cleveland, Ohio. Today I'm going to share with you our preclinical experience using PET imaging for hepatocellular carcinoma. To those of you who are not very familiar with a PET scanner, so this is the PET scanner with a double ring. Currently, there is no standalone PET. It will be either PET-CT or PET-MR because we want to register the PET measured physiology and biological information onto the anatomical structure. Currently, more than 90% of the PET scans were done with one radiopharmaceutical, showing here red as FDG. It is a glucose analog. At position 2, a hydroxyl group is replaced by radionuclide fluorine 18. So FDG will be transported into the cell just like glucose through glucose transporters. Then hexokinase will phosphorate into fluorine 18 FDG6 phosphate. And different from glucose 6-phosphate, it cannot go further into various biochemical pathways because the missing hydroxyl on position. So it is entrapped. It is the entrapment of metabolized radiopharmaceutical FDG over the time that gave us enough signal for PET imaging. During the radio decay of fluorine 18, 2 photons of high energy will emit in opposite directions, and the PET scan has no moving parts. It has a fixed detect ring which will catch these 2 lines, and then mathematical reconstruction will provide the in vivo distribution of the radiopharmaceuticals. For example, there is a small picture for a human whole body scan, and as you can see that the brain uptake is very strong because our brain metabolizes mostly with glucose, and on the arm there is some muscle tone and some of the others. And importantly, as I said earlier, PET imaging do not play a significant role clinically for hepatocellular carcinoma because in the liver, the metabolism of FDG is not outstanding for hepatocellular carcinoma, unless for polydifferentiate HCC. On the right is another scanner, a different imaging mode called SPECT, and it uses a different radionuclide for imaging, different from PET imaging, which we'll touch about. So to this audience, I do not need to emphasize that HCC is a global problem, although etiology varies from region to region. In Southeast Asia, southern China, it's mainly chronic hepatitis B. Here in the United States, it's hepatitis C. We've been working with this animal model of spontaneously occurring woodchuck HCC model. It is infected with its own woodchuck hepatitis virus, which is homologous to human hepatitis B. And when they got infected at birth, they went through acute to chronic hepatitis and eventually develop HCC. And today I'm going to show you some of the results from using this animal model. So the target for PET imaging is the oncofibroblastin extra domain B. From TCGA, it is shown that this extra domain fibronectin is a prognostic fact, and its expression level is higher in HCC in comparison to the surrounding liver. it is also highly conserved between the species. Showing here is the amino acid sequence, 100% identity between the human and the woodchuck. A specialized microarray was used to probe the expression level, showing that woodchuck HCC has higher expression compared to the surrounding liver parenchyma. And we also performed a PCR and Western blot from harvested woodchuck tissues to confirm this. Where are these oncofibrobacterians coming from? Well, they are shedded by fibroblast, but not your normal fibroblast. Rather, the cancer-associated fibroblast, CAFs, or tumor-associated fibroblast, TAFs. The CAFs or TAFs have been studied extensively, and here is a recent review written by the imaging people. The CAFs and the TAFs are in the tumor microenvironment, including in the HCC. And they have their own markers on the cell surface for targeting, such as FAP, which is fibroblast activation protein, and we will talk more about this. The interaction between cancer cell and the fibroblast back and forth allow the cancer cells to subvert those normal fibroblasts into CAFs or TAFs. And the shedded fibroblast will be further processed by MMPs and other enzymes to surround the cancer cells, or either as a barrier to the therapy, like drug resistance, or as a conduit to facilitate cancer cell migration and metastasis. So oncofibrobacterian was used as a target for imaging many years ago. So this study was published by an Italian group 20 years ago. And this is SPECT and CT. At the time, there is no combined scanner, so patient needs to be scanned separately. So they use antibody fragment single-chain FV, L19, labeled with iodine 123 for SPECT imaging. And showing here, panel A is the liver mets from colorectal cancer. B, C, D are metastasis from lung cancer. I want to try this for HCC. So at the time, I contacted the Italian group, and they said that Bracco licensed it. So I asked Bracco for 100 milligrams of L19, and they said that they are running clinical trial, and we have not heard anything from them since then. And we come up with a short peptide ligand targeting oncofibrobacterian. And from the selection going through phage display and molecular docking, and using molecular dynamics for binding affinity estimation. So the selection is not solely based on the affinity, rather taking into consideration of water solubility and stability. And before we use it for imaging HCC, we first tried on xenographic mouse models of pancreatic cancer. Showing here are two tumor cell lines, the xenographed model. And the image looks clean, especially look at the liver. So that prompted us to try this radiolabeled ZD2 short peptide for application PET imaging of HCC. Radiosynthesis of this radiolabeled ZD2 ligand is quite straightforward. So from the short peptide, linear short peptide ligand, we put a chelate node on it. And the final step, we chelate the radionuclide for imaging gallium-68. In addition, we also have DOTA labeled ZD2 and also another special chelate. So these are for imaging and also for commercialization purpose, which we will not talk about today. And here is the imaging results from using the Woodchuck HCC model. And showing here one is the coronal cuts PET-CT overlay. The other one is the axial cut. And the T here stands for the liver nodule HCC. H is the heart. L is the other lobe, the liver, free of cancer, and the stomach is mostly empty. We did a dynamic scan. As you can see from the curve that the tracer kinetics is rather quick. By 30 minutes or so, it's all stabilized. So meaning that after injection, you can just take a static image 30 minutes or later. After imaging, we did some validation with harvested tissues, both liver and the tumor. So as you can see on the up validation panel is that we use fluorescence labeled antibody G4 targeting fibronectin. And you can see the difference between the tumor and its surrounding liver tissues. And down the bottom is a convincing blocking study. So for our ligand ZD2, we labeled it with fluorophore. And as you can see that without G4 blocking, and the signal is strong. And with G4 blocking, there is no signal, meaning that the ZD2 radiolabeled ligand we use for PET imaging is specifically targeting the oncofibronectin. Remember our discussion about CAFs and TAFs. So our target, oncofibronectin, is a product of those CAFs or TAFs. But you can certainly image the CAFs and the TAFs directly because they have their own cell surface mark, FAP, fibroblast activation protein. And you can derive the inhibitors, FAPI, as imaging agent. And showing here is a recent example using FAPI-04 for imaging HCC. And HCC tends to be a cold spot, while the surrounding liver uptake is very high. And it is speculated to be liver cirrhosis. But histology only showed the tumor, not the surroundings. I expect the surroundings to show us the fibrosis. So here is another recent study using FAPI tracers targeting TAFs and CAFs for imaging hepatic sarcoidosis. It's quite interesting that the liver is not fully cirrhotic yet, and the imaging depicted activity of the CAFs and TAFs. Even more interesting is this another recent study where the same FAPI tracer used for imaging CAFs and TAFs are applied to cholangiocarcinoma. And as you can see that in comparison to clinically used FDG, the FAPI tracers show very strong uptake in the cholangioma, and the liver background is very low. There is no cirrhosis. So that gives us hope that for HCC applications, if the liver background is free of cirrhosis, we should be able to do it. And here is the comparison. So the same Woodchuck model of HCC was imaged twice, 2 days apart. And first, we use our ZD2 ligand labeled with gallium-68 for imaging oncofibronectin. And for the same animal, we use one of the FAPI tracer, also labeled with gallium-68. And as you can see that uptake is quite comparable. And I have to mention that the production of the radio tracers going through the same process using the same synthesis module. After imaging, we harvested tissue for histology and validation. Two samples here, both moderately differentiated. As you can see that in the surrounding liver tissue, there is no cirrhosis. And clinically, about 20% of the HCC developed without the background of cirrhosis. Half of them are chronically hepatitis B. So in a sense, our animal model represents this special group of patients. In conclusion, radio tracers for tumor microenvironment will be used for PET imaging of HCC, but only apply to a subpopulation of HCC patients without the cirrhotic background. Those FAPI tracers or our ZD2 ligands can be tested in future clinical trial. So with that, I will end. Thank you.